If all species of life on Earth entered a bodybuilding competition, who do you think would win? Leafcutter ants can lift 9 times their weight, and elephants are 5,000 kilos of solid muscle. But my money isn't on the animal kingdom. I think that plants would take the top prize because they literally build their bodies out of thin air. That's right, redwood tree trunks that soar 350 feet into the sky are formed by converting carbon dioxide gas into organic building blocks that make up plant bodies. Everything from the sturdiest branch to the most delicate flower petal to the juiciest fruit used to be carbon dioxide in the air. This process of taking carbon from CO2 gas and fixing it into sugars is called photosynthesis, and it's fueled by energy from the sun. Primary producers like plants and cyanobacteria are the only organisms on the planet capable of turning solar energy into chemical energy. Organisms that can't do this, like almost all other life on Earth, rely entirely on photosynthetic organisms to fuel their own lives. In this way, energy flows in an ecosystem from the sun to primary producers to organisms that consume primary producers either directly or indirectly. Understanding this energy flow in an ecosystem is crucial for studying everything from species diversity to climate change. Today, we're going to focus on the energy flow from the sun to primary producers. We'll measure the energy flow from producers to consumers in another video. So we'll be measuring just how good plants are at building their bodies, or how much sun energy are they converting into biomass. This is also known as net primary productivity. Net primary productivity is gross primary productivity minus the energy used for cellular respiration. In other words, it's the energy left over to grow and add biomass after plants have used what they need to fuel their own metabolism. For this experiment, you'll need aluminum weighboats, a digital scale with milligram precision, a drying oven, a lab notebook or spreadsheet for keeping track of your data, and a setup for growing Wisconsin fast plants. Wisconsin fast plants are a fantastic model organism for research because they grow quickly and are easy to maintain in a lab setting. They'll serve as the primary producers for our model ecosystem. We'll be growing about 40 plants for the experiment. Keeping lots of organisms together in a small space can sometimes lead to outbreaks of pests and disease, so it's really important to maintain a clean growth space, especially if you're growing multiple generations of plants in a row. First, let's sow our seeds. Each lab will probably have a slightly different setup, but generally, here are the steps for making your fast plants grow happily. Set up the containers you'll be sowing your seeds in. We're using plug trays. I'm going to fill them part of the way with soil, plop some slow-release fertilizer pellets in there, and then top them off with more soil. I'll soak the whole tray with water and then poke little depressions into the top of the soil with my fingers. Then I'll lovingly and carefully place one seed in each depression. Finally, I'll sprinkle soil over everything and wait for the seeds to do their thing. Wisconsin fast plants are fast, so it only takes a day or two for the seeds to germinate. They need bright 24-hour light, so you'll need to grow them under an artificial light source. They don't like it cold. I'm growing mine at 21 degrees Celsius. And of course, water them regularly. Yay, they're growing so fast! They're doing such a good job at primary production, taking in that light energy and using it to fuel the conversion of CO2 gas into carbohydrates. Go fast plants, go! After 7 days, we want to do a preliminary bodybuilding measurement. How much net primary production have our young seedlings done so far? We'll find out by measuring their biomass. Remember that in addition to the beautiful stems and leaves we can see, plants add biomass below ground to their roots. So when I harvest the plants, I'm going to be very careful to get the roots as well as the shoots. I'll harvest 10 plants in total, leaving 20 to 30 behind to keep doing their primary production thing. I'll carefully rinse the soil from their roots and blot them dry, and then place each one into a numbered aluminum weighboat to take their mass. Before I do, I'll place an empty weighboat on the scale and zero it so that the scale only measures the mass of my seedlings. I'm assuming that all the weighboats have about the same mass. I'll take the mass of all 7-day-old seedlings and record them in my spreadsheet. And all done, experiment over, right? Nope. We want to know how much organic matter our plants produced using energy from the sun, but in their present state, they have a ton of water inside their cells. We need to remove that water to get a dry biomass measurement. To the drying oven! The one in my lab is set to 60 degrees Celsius, but anything from 37 to 100 degrees will work. I'm going to place my wheyboats with their plants inside the drying oven, and now I just have to leave them there for 48 hours to completely dry out. 48 hours later. I'm going to take them out of the dryer using some oven mitts. Ooh, look how nice and crispy the plants are! After zeroing the scale with an empty wheyboat, I can mass the plants again. Now I have a dry biomass measurement for each plant. Let's analyze the data a little further. First, I'll calculate the mean wet mass and dry mass. At 7 days old, the seedlings had an average wet mass of 0.230 grams and an average dry mass of 0.024 grams. To get their percent biomass, I can divide their dry mass by their wet mass and multiply by 100. Wow, on average they're just 10.4% biomass. They're mostly water. But they still did a great job adding biomass for just 7-day-old little plant babies. Is there a way to depict their biomass in terms of energy? We would need to know how many kilocalories are in 1 gram of Wisconsin fast plants. And luckily, we do! The AP Biology folks have kindly provided us with a conversion factor of 4.35 kilocalories per gram of Wisconsin fast plant. If I multiply the average dry biomass, 0.024 grams, by the conversion factor of 4.35 kilocalories per gram, I get 0.104 kilocalories. And if I divide that by 7 days, it means these little guys were packing on 0.015 kilocalories per day. Champion bodybuilders in the making. Do older plants have a higher or lower net primary productivity? To find out, on day 14, I'm going to repeat these steps with 10 of my remaining plants. I'll harvest the shoots and roots, take the wet mass, dry them in the oven, take their dry mass, and analyze the data. This time, their average wet mass is 2.763 grams, and average dry mass is 0.347 grams, meaning they're 12.8% biomass on average. Still mostly water. The average dry mass of 0.347 grams multiplied by our conversion factor of 4.35 kilocalories per gram divided by 14 days gives us a net primary productivity of 0.12 kilocalories per day. This is 8 times higher than the younger seedlings' net primary productivity, and the 14-day-old seedlings packed on most of that biomass after they were 7 days old. Gosh, I need to start lifting if I want to keep up with them. Now that you know the methodology for measuring net primary productivity in plants, you're all set to learn how to measure secondary production as consumers, well, consume the primary producers. Sorry, plants. We'll cover this in another method video. You'll eventually go on to design your own energy flow experiments. So I'd also start thinking about what parameters you could change around the sun energy flowing in or the productivity of the photosynthesizers and how those might alter the energy budget of an ecosystem. In other words, how might you enhance or inhibit the bodybuilding abilities of your fast plants?